Welcome to Study Loop.
This is the answer to Chapter 21, problem number 81 from the Smith Organic Chemistry textbook. This problem gives us 4 sets of data and asks us to come up with a structure for each set of data. And so I'm going to jump right in because this is quite a large problem. Okay, so the starting point for each of these is going to be to calculate an HDI based on the formula that we're given. Remember, that's going to be 2 times the number of carbons, which in this case is 5, plus 2 minus the number of hydrogens, which in this case is 10, and all of that over 2. So we have 12 minus 10 is 2, over 2 is 1. So we have 1 degree of unsaturation in this molecule. So 1 double bond or 1 ring. Looking at the IR data, we have a peak at 1728. That is going to be indicative of a carbon-oxygen double bond. And then the other 2 peaks that we have between 2700 and 2791, that is going to indicate an aldehyde. So that's our 1 degree of unsaturation. So then looking at the NMR signals, our second signal at 9.48 ppm with an integration of 1 is very far downfield. And so that is indicative of an aldehyde. Okay, and we only have one other signal, 1.08 ppm singlet with an integration of 9. So this is going to be 3 methyl groups. So 3 CH3s. And so that's 4 carbons accounted for. So I'm going to say that what we have here is a central carbon with 3 methyl groups on it. And then an aldehyde. Okay, so there's our structure for A. So now we need to do this for the other 3 sets of data that we have. So for B, the formula is exactly the same as A, so we don't need to calculate the HDI. It's going to be exactly the same as A's was. So again, our HDI is going to be 1, so 1 ring or 1 double bond. Our only IR signal is at 1718 wave number. So this is, again, indicative of a carbon-oxygen double bond. I'm going to say probably a ketone this time. And then looking at our NMR data, we have a 1.10 ppm doublet with an integration of 6. And so this is going to be 2 equivalent methyl groups. And they're going to be next to a carbon with a single proton on them, which is why they're split into a doublet. And so they are the first and third signal are splitting each other, basically. And so we know that our third signal is going to be a piece that looks like this. So we have 2 methyl groups, and then a bond to something else. And then the signal is actually coming from a hydrogen right here. So we have these 2 pieces figured out. And then we have a singlet at 2.14 ppm that integrates to 3. And so this is probably another methyl group. Okay, and so when we put all of this information together, it kind of has to look like this. So here's our methyl group. Our ketone is going to be right here. And then we have the rest of the molecule here. Okay, and so that's the structure for B. So moving on to C, we need to calculate an HDI here. So for this formula, it's going to be 2 times 10, it's the number of carbons, plus 2 minus 12, which is the number of hydrogens. And so 22 minus 12 is 10, divided by 2 is 5. So we have an HDI of 5 here. So right away, anytime I see 4 or more, I'm thinking an aromatic 6-membered ring, since that accounts for 4 degrees of unsaturation. And let's jump to the NMR. So 7.24 ppm doublet that integrates to 2. So this is probably 2 aromatic protons. And the last signal at 7.85 ppm, also a doublet, also an integration of 2. That's going to be 2 more aromatic protons. So we have an aromatic ring here, and it's disubstituted. And it's probably para-disubstituted based on the fact that our signals are both nice doublets with integrations of 2. So looking back at the IR, that peak at 1686 wave number, that is going to be indicative of a carbon-oxygen double bond. So we have a ketone here, and I'm going to say it's definitely a ketone because we don't see an NMR signal that corresponds to an aldehyde. So this is definitely a ketone. So looking back at our NMR, our first signal integrates to 3. It's at 1.21 ppm. It's a triplet. So this is probably a methyl group. Our next signal at 2.39 ppm is a singlet. Also integrates to 3. So this is probably a second methyl group. And this is pretty far downfield for a methyl group. So I'm thinking one of the substitutions on our aromatic ring is just that methyl group. So I'm going to go ahead and start to draw the ring. And then here is our methyl group that our second signal is telling us about. Okay, and then our third signal, 2.95 ppm quartet that integrates to 2. So this is going to be a methylene group next to a methyl group. And we know that because it's split into 4 peaks, which means it has 3 neighboring protons. And so the only way to put this together so that it makes sense is going to be to put our ketone right here next to the ring. And then here's our methylene group that the third signal is telling us about. And here is our methyl group that our first signal is telling us about. And so that's the answer to C. And so lastly, for part D, our formula is the same as part C. So our HDI is going to be the same as part C. So once again, we have an HDI 5. So again, I'm thinking aromatic ring. Looking at our NMR signal, the fourth signal, the 7.06 to 7.48 ppm multiplet with an integration of 5 tells us that we do, in fact, have an aromatic ring and that it is monosubstituted. And so we know that because that's the chemical shift range for aromatic protons, an integration of 5, and the fact that it's like a messy multiplet. Those are all good indicators. So we'll say mono substituted aromatic ring. And we know it's mono-substituted because the integration is 5. Okay, so our IR, once again, tells us that we have a carbonyl, carbon-oxygen double bond, and that's all it tells us. So looking at our NMR signals, the first signal, the 1.02 ppm triplet, integrates to 3. So this is a methyl group. And then we have a 2.45 ppm quartet, and that integrates to 2. So this is probably a methylene group, and it's a methylene group next to a methyl group. And again, we know that because of the splitting. So it's split into 4, so it has 3 neighbors. So signals 1 and 2 are splitting each other. So we can tell that. And then the third signal is a 3.67 ppm singlet that integrates to 2. So this is going to be another methylene group. And because it's shifted so far downfield, I'm going to say that the structure is this. So we have our ring, we have our first methylene group, and it's shifted so far because it's attached directly to the ring, and also because it is attached directly to the ketone that's right here. And then we have our next methylene group, so the signal number 2, and then our first signal, the methyl group, is right there. Okay, and so that's the structure for part D. Okay, And so again, these were all solved by doing an HDI first, considering the evidence from the IR, and then mainly by using NMR to figure out what pieces we had and how the pieces all fit together. And that's the answer to chapter 21, problem number 81.